Hello students, in this video I'm going to discuss about variations and Mendel's monohybrid experiment and Mendel's first law of inheritance. Now what are variations? Variations are differences. In this living world, all the living organisms are so different from each other. Even if we consider a particular species, the members of a particular species have common body design, but still there are so many differences between them. No two members of a particular species is exactly same. What is the source of this variation? Reproduction. In asexual reproduction, the offsprings get the exact DNA copy of the parents. In asexual reproduction, in vegetative reproduction, the offsprings are copy of the parents, but still some differences are there, and the source of this difference in asexual reproduction producing organism is the errors during DNA copying. The errors produced during DNA copying results in very small amount of variation, which is insignificant. The major source of variation is sexual reproduction. In sexually reproducing organisms, diploid organisms having two sets of chromosomes, they receive one set of chromosome from the male parent or the father and the another set of chromosome from the female parent, that is the mother. Sexual reproduction is the main reason behind variation due to inheritance. Then there are other sources of variations. There are environmental reasons for variations. Then there is mutation, etc., etc. These are the other sources of variation. Next important question is, are variations useful or variations are harmful? Yes, variations may be useful or harmful. Let's consider the example of bacteria. Suppose some bacteria are heat resistant. During heat waves, the heat resistant bacteria will survive, and those who are not heat resistant will be wiped out. In this case, the variation is useful for the survival of the bacteria. There could be some harmful variations that may result in death and extinction of the organism. So, and another important or significance of variation is that variation helps in evolution. Due to variation, new species are evolved from the pre-existing species. Now, what is inheritance? Inheritance is what is given, what is passed on from the parent to the offspring. That is called inheritance. For his plant hybridization experiments, Mendel selected the pea plants. The reason behind this may be pea plant can be easily cultivated. It has a very short lifespan. It completes its life cycle in a short span of time. Thirdly, it has bisexual flowers. And fourth reason is that it has 7 pairs of contrasting characters like height, tall and short, position of flowers, axillary or apical, shape of the seeds, round, wrinkled, color of the seeds, yellow, green, etc., etc. There are 7 pairs of contrasting characters. In his first experiment, Mendel's first experiment is called the monohybrid experiment. In monohybrid experiment, Mendel only considered 1 pair of contrasting characters like height, tall and short. In his monohybrid experiment, Mendel selected 2 plants having opposite traits. like a pure tall plant and a short plant. Short plant is always pure. In his monohybrid experiment, Mendel selected a tall pea plant and a short pea plant as parents. And this generation is named as the P1 generation. After a cross between pure tall and short plant, the new offsprings that are produced are named as the F1 generation, and all the plants in the F1 generation were tall plants. None of the plants were short. So, as in the parental generation, there were two characteristics of the parents. One parent was tall and the other parent was short. But in the F1 generation, only one of the characteristics was expressed. So, the characteristics that is expressed in the F1 generation is called the dominant characteristics. So, in height, the tall is the dominant characteristics. Similarly, in color of seed, yellow versus green, yellow is the dominant characteristics. In the shape of the seed, round versus wrinkled, round is the dominant characteristics. Position of the flower, axillary versus apical, the axillary flower is the dominant characteristics, etc., etc. So there are 7 dominant characteristics in Mendel's pea plant or pea plant. So, and then what happened next step? All the tall plants produced in the F1 generation are then selfed. So the F1 plants were selected as the parent for the next stage, and they were named the P2 generation. And the plants obtained in the F1 generation were named P2, and they were selfed, means they were used as parents for reproduction. The P2 generation produced offsprings, and those offsprings produced by P2 generation were named the F2 generation. In the F2 generation, we found that out of every 4 plants produced, 3 were tall and 1 is short. The ratio between tall and short plant in the F2 generation is 3:1. So what we find that in the F1 generation, all plants were tall. The short characteristics was almost lost. But in the F2 generation, we again found that out of every 4 plants, 3 became tall and 1 became short. So the ratio between the tall and short was 3:1, and this 3:1 is considered as the F phenotypic ratio. Phenotypic ratio is based on the outer or exterior characteristics. And the genotypic ratio of monohybrid cross is 1:2:1. Here, for every 4 plant in the F2 generation, 1 is pure tall, 2 is heterozygous tall or hybrid tall, and the 1 is short. So how a plant is pure or heterozygous, how it can be found out whether a pea plant is pure tall pea plant or it is hybrid or heterozygous tall. It can be ascertained by a method called test cross that you will read later. And the short plant is always pure. It is recessive characteristics and it is always pure. Short plant is always pure. We get 2 laws from Mendel's monohybrid experiment. The first is the law of dominance. It is better to call it a corollary rather than a law. According to law of dominance, in a pair of contrasting characters, one of the characteristics is dominant and the other is recessive. The characteristics that is expressed in the F1 generation is the dominant characteristics, like the tallness, second, the round shape of the seeds versus the wrinkled shape. The round is the dominant. The yellow colored seed is dominant and the green colored seed is the recessive, etc., etc. The second law that we get or from the Mendel's monohybrid experiment is called the law of segregation. Law of segregation is better known as the Mendel's first law. As the law of dominance is not considered as a law, it is in several books, it is considered as a corollary, not as a law. So law of segregation is the first law of Mendel that is obtained from the monohybrid experiment. According to the law of segregation, the alleles separate during formation of gametes, and they are randomly goes to the gametes, and the alleles separate during the formation of the gametes, and this is called the segregation, law of segregation. What is meant by pure or homozygous or hybrid or heterozygous? What is mean by all this? Pea plant has 7 pairs of chromosomes. Each chromosome has 2 copies, and it is a diploid organism, and each chromosome has 2 copies, and the 2 copies of a chromosome is called homologous chromosome. And in the locus of the homologous chromosome, if the same allele is present, allele is a form of the gene. Like for height, there are 2 forms of gene, tall and short. If in the homologous chromosome tall allele is present, allele for tallness is present in both the chromosomes, this plant is called homozygous tall or pure tall plant. And if in one of the homologous pair allele for tallness and the other pair the allele for shortness is present in the locus, then it is called the heterozygous tall. And similarly, if short characteristics is expressed when allele for short is present in both the locus of the homologous chromosome.